Kennedy.
Okay, good evening. Tonight I'm going to give you a short lecture on the structure and the universal structure of DNA and chromosomes. DNA and chromosomes are basically made up of components, the basic components which are known as nucleotides. So the very monomer unit of nucleic acids are nucleotides. And nucleotides are made of three components. These are the sugar, the phosphate, and the nitrogenous base. For the sugar, it's either the deoxyribose sugar for DNA or ribose sugar for RNA. And for the nitrogenous bases, there are also four types. There are four kinds or classes. You have adenosine, guanosine, cytosine, and thymine. And these are grouped into either purines and pyrimidines. Purines are composed of adenine and guanine, while pyrimidines are composed of cytosine and thymine. Now, added to the thymine, to the cytosine and thymine is uracil, because there is no thymine in RNA. Thymine is only a component in the DNA. So let's try now to look at the global structure of the DNA. So now we are going to discuss the universal structure of DNA and chromosome. So basically, DNA is the basic unit or the basic structure of a chromosome, which are composed of the nucleotides. So when you put the nucleotides together, you call it polynucleotides. And so let us try to look at the DNA. So first, we have to compare the DNA between prokaryotes and eukaryotes. Prokaryotes are represented by bacteria, and eukaryotes are example is the human chromosome. The difference between the two is that in prokaryotes or in bacteria, it is simple, it is circular, there are no introns, no histones, and they are directly found within the cytoplasm. Whereas in eukaryotes, it is complex, it is linear compared to bacteria, which is circular, with introns, and introns are known to be intervening sequence with no equivalent genes at all. In other words, introns are intervening sequence that are found in DNA that does not code for any protein at all. It is not also, these introns are nonsense codes. So the opposite for that is the exon, which is the code. So the all genes are exons, whereas those that are in between are introns or nonsense codes. And you know what? In humans or in eukaryotes, there is an excess of junk DNA. You call it introns. It is estimated about 90% of the DNA in eukaryotes are nonsense codes, or 90% of the DNA codes in eukaryotes or in humans are nonsense codes. So these nonsense codes are sometimes we think that they are useless, but in essence, nonsense codes are protective mechanisms by which the DNA of the human is protected from possible damage. So on a daily basis, if you're exposed to sunlight, and since sunlight are deleterious and damaging to DNA, at least when you're exposed to sunlight, 90% of the time, the one exposed or damaged are nonsense. In other words, it does not affect the whole structure, because after all, that 90% is nonsense, it is blank, and therefore proteins will be protected, possibly protected. So for me, the 90% is a protection against radiation damage. So somehow, there are some good explanation, good implications for this, that the 90% nonsense codes are protection against possible DNA damage. So these introns are nonsense, and those exons, which are genes, are wound up in a protein called histones and non-histones to organize them into chromosome. And once the histones are, or all the DNA that are wound up in the histones are condensed, they are known as chromosomes. And the chromosomes are sequestered in the nucleus of the cell. That's why it is sequestered. So you can see here, the DNA is sequestered inside the nucleus. So this is the structure. So you have the nuclear envelope and the nucleolus, which is part of the nucleus, and the rest are DNA materials. So you have seen here the nucleolus. This is the nucleus. So the one that you see inside, the dots that you can see inside the nucleus are the DNA materials. You call it heterochromatins. So the DNA is sequestered in the nucleus. It's about 10% of the total volume of the cell. So the DNA comprises 10% of the total volume of the cell. It is determined by nuclear envelope, and these envelopes are punctuated with nuclear pores. So there are pores. In bacteria, it is direct. So this is actually the caricature of the structure of DNA. It is a double-stranded, either left or right, or one side or the other side, and it is arranged in a helical structure. So this is how the DNA looks like. It is composed of the bases. These are this one, guanosine, or paired with cytosine, and thymine paired with adenine. So they are what we call complementarity. There is a complementarity, which is a constant comparing between purine and pyrimidine. A purine adenine is paired with a cytosine thymine, or a purine guanine is paired always with cytosine. So given there are free nucleotides, once they are placed in water, the nucleotides will seek its pair naturally. You call it natural complementarity. G will always look for C, and A will always look for T. That's why that is what we call the cohesive property of the nucleotides. And the one that binds between them are what we call hydrogen bonds. So the helical structure of the DNA is composed of there is what you call the backbone of the molecule. The backbone of DNA molecule is composed of sugar phosphate backbone. Sugar phosphate backbone is a very strong structure, because the sugar phosphate backbone is composed of the phosphodiester covalent bonds, which are not even broken by heating. However, in heating, the hydrogen bonds between the nucleotides, the base pairings, are melted or are broken. So this is the structure. This is the phosphodiester bond. This is the backbone structure. And then we have the nitrogenous, which is adenine, always paired with thymine. So adenine paired with thymine has 2 hydrogen bonds. Guanine paired with cytosine has 3 hydrogen bonds. And I told you, these hydrogen bonds can be broken by heating. That's why DNA could be melted at the hydrogen bonds. If you heat DNA, it will break the hydrogen bonds. But if you heat DNA, the phosphodiester bond, which is the backbone, is not melted, or it's not broken by heating. So in other words, DNA is stable, and it cannot be broken by heating, the primary structure of the DNA. So this is how it looks like, the pleated sheets, the helical structure. So this is another representation of a double helix of the DNA. And this is another view of the hydrogen bonds between the bases. So most chromosomal DNA codes does not code for proteins. So as I said, 90% of the DNA codes are introns or nonsense codes. The genomes of higher organisms to contain large excess of nonsense codes. Each gene produces on DNA. Histones are the principal protein that binds the genes together. So to organize a gene, they are organized always by a protein called histones and also non-histones in order to compact them into one domain. So this is it. So this is the DNA and these are the histones. So in other words, the DNA is always wound up into a histone. This is what it looks like. So these are histones and the DNA is wound up into the histones. And accordingly, each histone holds one gene. So it is very logical that histones are very important in making the DNA stable or the gene stable. So this is how it looks like in the electron microscope. These are the chromosomes, and the beads that you can see there are what we call the nucleohistones. These are actually the heterochromatins, that's it, the beads that you can see. So one like that is about 30 nanometer. So the location of the nucleases, histone 1, histone 3, H3, H4, H2A, H2B. So in other words, one nucleosome has 4 histones. What are these 4 histones? H2A, H2B, H3, and H4. And once they are packed together, these 4 histones are attached to, are being what we call annealed to by the protein called histone 1. So we can actually classify 2 groups of histones inside the chromosome, the H1 histone and the nucleohistones. So these 4 histones, H2A, H2B, H3, and H4 are known as nucleohistones, whereas the other type is H1 histone. So H1 histone is a separate classification of histone. So these are the amino acid sequence of the H4, and this is the histone, and there are some linker DNA. So you can see these are now the linker DNA between one histone to another histone, nucleohistones. So if they are, once they are, so you have a DNA, and then it is wound up into a histone. It will make about 11 nanometer. So it is octaneric H2A, H2B, H3, and H4. So this is, now once his nucleostones are packed also in another domain, it leads to another level of complexity. We call it the 30 nanometer chromatin fiber. It is again, so in one fiber it has about six nucleosomes, yeah, like that. So it will make into a this is what we call a 30 nanometer fiber, and in between are what we call DNA binding proteins, which are specific for a specific type of chromosome or genes, set of genes. So when that 30 nanometer fiber are also arranged in the non-histones, they will form into another domain. Chromosome is then shown to be folded further into that kind of structure, and it's about that much. So this is the histone 1, these are the nucleohistones. So the histone 1 are the ones that connect one nucleosome to another nucleosome. So this is how it looks like. So these are the chromosomes or oocytes. So these are the global structures of the DNA in humans. It is arranged from the largest chromosome 1 to the smallest chromosome X and Y. So since we have 22 pairs of autosomes and 1 pair of sex chromosomes, that is comprising now the 46 chromosomes, because 22 times 2 is 44, plus X and Y is 45, 46. So this is actually the global structure of once the chromosomes are organized into into specific domains, they can be further grouped into 3 parts. We call it the telomere sequence, the replication origin sequence, and the centromere, as well as the other parts of the chromosome. So this is how the chromosome looks like. So from simple double-stranded DNA, which is 2 nanometer, once it is folded by histone, it becomes 11 nanometer. And this 11 nanometer is packed furthermore, it becomes 30 nanometer. And this 30 nanometer are packed again further, it becomes about 700 nanometer, 300 nanometer. And then we have the 700 nanometer up to about, pila na siya?
1.4.
1.4 nanometer. So this is now the tightly packed chromosomes. So the one that pack the chromosomes are, so when you are going to define it, a DNA are just simply nucleotides. A gene are nucleotides with corresponding proteins. And a chromosome is a gene plus DNA plus protein is known as chromosome. So when you talk about chromosome, it looks like already a fully packed structure with the center, which is a centromere that holds the 2 daughter cells, 2 daughter strands rather, and then you have the chromatin. So there are 3 specific sequences in chromosomes. We call it the replication origin sequence, and the centromere that holds the chromosome at the center, the two daughter chromosomes, and we have the telomere sequence. So these are 3 specific sequences in the chromosomes which are very distinct during the condensed state of the chromosome. Now, why is it very important to pinpoint that there is what we call centromere? The centromere is the attachment of the chromosome during the metaphase stage of the cell cycle, because the chromosomes are aligned at the center, and it is through the centromere where the microtubules are attached and causes the mobilization of the chromosome either to move away or to be pulled together. So through the centromere. And the replication origin are DNA sequences by where the DNA is duplicated. Now, what is the significance of this replication origin? The significance of this replication origin is that if there are no several clusters of replication origin, it would take about 800 hours to duplicate a DNA from one end to another end, because it was found out that duplication of chromosome or DNA takes about 500 nucleotides per second. So, granting for the sake of argument that there are 150 billion nucleotide base of one chromosome, 150 million, you divide it by 500 nucleotides per second, it would take about 800 hours from end to end. But it is impossible for the cell to duplicate, requiring 800 hours every time the cell divides. It's very impossible. Because 800 hours, if you divide it by 24 hours in a day, it would take 33 days, which is very impractical. Our cells divide every now and then, and it is impossible that it will only divide after 1 month or 33 days or 300. So we have that. So these sequences are relatively short and less. About the 3 types of sequences elements are thought to operate in human chromosome, but to date only human telomere sequence have been well defined. So this is the human chromosome in karyotype. This is called the karyotype. The bands that you can see there are specific locations of the genes. The bands that you can see there. So our DNA are organized into domains, and these are what you call the clusters of genes or you call it pair of genes in an allele. Or you call it in genetics is called the locus. The location of the gene in the chromosome is called as the locus. The pair of gene is defined as allele. So the DNA, allele, chromosome, genes should be delineated by definition. So this is how it looks like. So chromosomes are organized in several clusters of there are different genes in a chromosome. So it was thought to have there are about more than 30,000 genes in the human body. And these 30,000 genes are arranged in different clusters in different chromosomes. So the 30,000 genes are scattered in several chromosomes. So one cluster of gene is found in another chromosome. Foremost example I would like to cite here is the protein hemoglobin has two clusters, the alpha cluster and the beta cluster. The alpha cluster is found in chromosome 16, while the beta clusters of the globin are found in chromosome 11. So in other words, every part of our body are proteins which are assigned in different chromosome numbers. So these are the three sequences. The set of entire chromosomes. So these are what you call the bonding patterns. That one you see in bonding patterns. The bonding patterns. That's all for the global structure. The DNA.